Hello everyone, we'll start in a few minutes. Okay, I think we can start. Is my audio audible? Is my voice audible? Can anyone let me know? Hello? Yeah. And if my presentation is not visible or if I am not audible, you can please turn on your mics and let me know, because after going into the presentation mode, I will not be checking the chat boxes. So please turn on your mic, and if you want to ask any questions or if you have any doubts or if my presentation is not visible, you can turn your mic on and tell me. Okay, I hope my presentation is visible. So yes, does anyone want to tell anything? Okay, so welcome to Host Pathogen Interactions live session. This is for week 3.
Presentation is not visible, ma'am.
Okay, thank you. So I'm Sumati Raviraj from University of Hyderabad, PhD scholar, and the course instructor is Professor Himanshu Kumar from ISIL. So as these are the important dates, uh, kindly register for the exam and fill in the forms for exam registration so that you will get a certificate if it's required. Okay, so this week the syllabus was cells of the immune system and its role in host defense. So in this, there are many topics that would have been covered in the videos. The PBMC isolation, neutrophils, eosinophil, basophil, mast cells, what are their roles, what is their functions, dendritic cells, macrophages. Somebody's mic is on. Can you please mute it?
Ma'am, your presentation is not visible, ma'am.
It's not visible.
Yes, ma'am.
Uh, okay, one second. Is it visible now?
Yes, ma'am.
Okay. Yeah, thank you for informing me. So, as I was saying, in this week, the syllabus that was covered was cells of the immune system and its role in the host defense. So, there you would have had the videos for PBMC isolation, functions, features, and roles of neutrophils, eosinophils, basophils, and mast cells, and dendritic cells, macrophages, natural killer cells, NKT cells, B and T cells. So, coming to the assignment. So, we will be going through few topics which are related to the questions in your third week's assignment. So instead of giving the answers directly, we will just understand a little bit basic about each question, so that this will help you answer your questions over there. Okay, so let's start with the first one, that is neutrophils. So neutrophils play a crucial role in the acute inflammation as the first responders of the immune system. So the first responders are the ones that reach the site of infection or reach the site of injury immediately. That is their most important and crucial role. That's why they are called as first responders, because they are the first cells in the body to respond to any sort of infection or injury. Okay, so that is very important to remember. And their primary functions include rapid recruitment. So because they are the first ones to arrive at the site of injury or infection, their recruitment should be very rapid. So they can reach to the site of infection as early and as soon as possible. Okay, because the infection or the injury must be cleared quickly. So these cells should be able to recruit at those places quickly as soon as possible. So rapid recruitment, neutrophils migrate to the site of infection or injury in response to chemotactic signals. So there should be some sort of mechanism by which these cells will be able to recognize or will be able to move to that site of injury, because there should be some sort of signal for them to be recruited over there. So these signals are the chemotactic signals. So these are nothing but your cytokines. Those are your IL-8, interleukins, complement C5a, LTB4, etc. So when your injured cells or your infected region in your body, these cells will secrete certain signals that are your cytokines and chemokines, which will attract your neutrophils. So these chemotactic signals help your neutrophils to recognize and identify the site of infection and site of injury and reach the place immediately. Next function is phagocytosis. Phagocytosis is to engulf and digest the pathogens. So they engulf and digest the pathogens, dead cells, and debris. So once they reach the site, their role is to phagocytose the pathogen. So you can see here in the picture, phagocytosis is one of the key features of this. So it will engulf directly the pathogen and it will digest the pathogen within the cell and discard it and stop the infection. Degranulation. The next is degranulation. So neutrophils release granules containing enzymes like myeloperoxidase, elastase, and lysozyme to break down the pathogen. So this is how the pathogens will be degraded or broken down using various enzymes which are released in the form of granules. So this process of releasing these enzyme containing granules is called as degranulation. Next is ROS production, that is reactive oxygen species. One second. Yeah. So the reactive oxygen species, they generate the neutrophils, they generate a lot of ROS, and via the enzyme that is NADPH oxidase system, these help to kill the microbes by increasing the ROS levels in that particular region. Then neutrophil extracellular traps are also called as nets. So they release these neutrophil extracellular traps which literally trap the pathogens and neutralize them. You can see here, this is netosis. So it's like a net which will trap the pathogens, and then these pathogens will be neutralized. That is nothing but broken down and damaged here. So this is extracellular mechanism. Next, pro-inflammatory cytokine release. So neutrophils secrete cytokines Neutrophils themselves also secrete cytokines. Like we saw, there were several cytokines that were released here. So the interleukins, etc. These are by the damaged and injured cells for the recruitment of your immune cells. But your neutrophils themselves also recruit several cytokines after reaching the site of infection to amplify your immune response. Okay, so it will recruit further more other types of immune cells which are required for that particular type of infection or that particular type of injury. Specific cells will then be again recruited. So they also release TNF alpha, IL-1 beta. So depending on the cytokines that are released, different signals will be activated and different type of immune responses will be amplified. So this is your basic for neutrophils. So the major points that you have to remember about neutrophils is they are the first responders, and they attack the pathogen by phagocytosis, netosis, that is neutrophil extracellular traps. Then they have degranulation mechanism in which they have the enzymes to degrade the pathogens. They also release a lot of ROS, which help in uh, damaging the pathogens as well. And then they also produce several cytokines to activate or amplify the immune response further, depending on the type of response that is required. Next is, yeah, and also their activity is short lived, and once the infection is controlled, they undergo apoptosis and are cleared by macrophages to prevent excessive tissue damage. So since these cells are mostly pro-inflammatory kind of responses, so every immune response has to be regulated, right? So these once the inflammation or the infection has been controlled, the damage uh, to the pathogens or any kind of injury has been completed, then these cells should now be stopped. So then now your macrophages and other cells come into play. They will stop your neutrophils. They will clear the neutrophils to prevent your excessive tissue damage. Otherwise, tissue damage or injury can also occur if your neutrophils are not controlled at proper time. Next. So next was about the syngenic mice model. So the syngenic mice model refers to an experimental animal model in which the mice are genetically identical or nearly identical to each other. So these mice are generally inbred mice. So that is the reason they are genetically identical. So this means the mice used in the model are of the same inbred strain. So after continuous inbreeding, you will get the inbred strain. So this is done to avoid heterogeneity in the mice population. So you will get a similar type or more identical type of mice. So and they typically have a uniform genetic background. Then syngenic mice models are particularly useful for studying disease processes in immunological studies, in cancer research, because they minimize the genetic variability, allowing researchers to focus on the specific experimental manipulations and treatments. So as the heterogeneity increases, your effects might not be same as you see in each mice or each strain of mice. So to avoid this sort of variability in your results, because reproducibility and similar result, statistical significance, etc., is all very important to establish some kind of discovery or some kind of experiments that you are doing. So for this, to avoid variability due to the animal itself, the generally inbred mouse are taken, so that your variability due to the response which the animal gives because of the heterogeneity in its genetic background will be decreased. So the key characteristics and uses of syngenic mice model are: first is genetic homogeneity, that is when they are continuously inbred, they will be more homogeneous to each other, and they will be more genetically identical. So the mice of the same inbred strain, such as C57 black mice, are used generally in most of the researches, because the reproducibility of these experiments are generally high with these strains. So ensuring that all the animals have the same genetic makeup, so because of this they will give a similar kind of response. This helps eliminating variations in immune responses or disease progression that could result from genetic differences. Okay, then next is they are also very useful in tumor model as tumor models. Syngeneic mouse models are commonly used in cancer research, where the tumor cells from a specific mouse strain are implanted into other mice of the same strain. Since the donor and the recipient are genetically identical, the model avoids graft versus host disease. This we discussed in the previous session also. So, graft versus host is one of the main immune responses which does not allow such kind of experiments, or will lead to failure of experiments. And also, if you're really doing a graft, a therapeutic graft research or a transplantation research, then this will not work if your mice strains are not identical. They will generate immune response and they will reject it. So this can be avoided by using syngenic mice model. So the model avoids graft versus host disease, providing insights into how the immune system interacts with the tumors. So to study the interactions of the immune system with the tumors, we need the host to first accept that tumor and not immediately reject it. So when your tumor is implanted from a different mice to another mice of the same strain, it should not reject it, so that your immune response will be generated, whatever treatment or whatever drug you want to use against that tumor. If you want to see how the mouse's immune system is responding to the drug and reacting to the drug, or helping the drug attack the tumor, all these kind of things can be done. Then immunological studies. These models are ideal for studying immune response in a controlled environment, like we discussed in the previous session. So in a controlled environment, that is, sometimes we use this, we give irradiated mice for several experiments. So that time you don't want the host immune system to, affect it, whatever uh, you are injecting into the mice. If you want to see one specific effect of one specific type of drug or one specific type of immune cell response, then you don't want a complete system in between, because the variables increase. So if you want a very specific study, then these kind of mice are very good model. So, for example, researchers can study the immune system's response to pathogens or how it behaves in autoimmune diseases without the confounding factors of genetic diversity. So, when there is genetic diversity, even if you're studying a very specific immune response, each mice might react or respond in a different way based on its genetic makeup, because few of the genes in one particular mice might help generate a different type of response, or it might amplify more than the other mice. So all these kind of variabilities can be eliminated. Then transplantation studies, like we said, because the immune system of the syngenic mice recognizes the transplanted tissue as self, it will not reject it, it will recognize it as self. So the syngenic mice models are used to study tissue graft survival and rejection mechanism through though typically without rejection due to genetic matching. So if you want to study this transplantation rejection survival, tissue graft survival also, this is very useful, because mostly it will not reject the tissues immediately. Then if you want to see one specific, if any specific gene activation, or if any specific gene is affecting or causing rejection, you can then knock in that gene, or you can, induce some rejection mechanism and check whether if it is through that mechanism, all these kind of studies. So it is very helpful in studying your transplant tissue graft survival. Next is standardized experimental conditions. Using syngenic mice reduces the variability caused by genetic differences, which makes experimental results more reproducible and reliable. So this is one of the major things. Reproducibility and reliability increases when you use your syngenic mice model. So in syngenic mice model, since the genetic similarity is very critical, because these models involve mice that are genetically identical, typically from inbred strain. This genetic uniformity plays several roles in different areas. So one major thing is your immune compatibility. Since the mice share the same major histocompatibility complex, so your MHCs are same genetically, because your MHCs also differ in your population. Even if you belong from the same species, your MHC classes differ, and based on your genetic makeup, based on your paternal and maternal side. So this kind of MHC compatibility is done in syngenic mice. So there is no immune rejection of the transplanting tissue. So MHC is the main which causes your transplant rejections. So if it is non-compatible, it is then easily rejected. So here it is immune compatible, that is your MHC compatible. So this uh, either for your transplanting tissues, your tumors, your cells from one mouse to another mouse, anything, but all within the same strain of mice. Then this makes the syngenic model ideal for tumor biology and immune response. Then reproducibility. Genetic uniformity minimizes the variability of experimental outcomes, allowing for consistent and reproducible results in studies related to drug efficacy, disease progression, and immune responses. So why this reproducibility and variability is very, very important in case of your experimental studies is, uh, like generally in research we do that, we take, when we take mice models, we usually have n, that is the number of mice in which your same kind of experiment, your same exact same experiment is done. If for example, if you want to test the efficiency of a drug, suppose drug A, and if you want to test compare it with drug B, so you have to have two groups first of all, and you need to then have another control group also. You can have several other control, but one control group is main, in which you won't give any drug, and then one group with your drug A and one group with your drug B. So if you take two or three mice, and then start your experiment, inject the drugs and check the efficacy. Maybe it's against tumor, to decrease the tumor, shrink the tumor, kill the tumor cells. So if you see in maybe in drug A was able to suppress or able to shrink the tumor in two of the mice, but not in the third mice. In case of drug B, it was able to shrink it in two, and again not in one. So you might think that since both of them are affecting uh, in two of the mice in each group and not in one mice, they both have similar efficacy. But when you increase your number of your mice, that is when you increase your n, if you take it to 6 or if you take it to 12, minimum we generally go for n equal to 6 when we do with mice model. So when you go to 6, then you might see that uh, 4 out of all the 5 or 4 out of all the 6 in drug A are accepting the drug, and the drug is very efficient out of the two mice. Or in case of drug B, out of the 6, 5 are taking up, and 5 mice it's more efficient. So when you increase your number of mice, you will see your statistical significance will increase, and then you can see maybe that one mice in that group, due to some other reason, it could not have taken up the drug, the drug would have not been helping the tumor to shrink. So this could happen in case of your heterogenetic mice, where the genetically they are not really identical. because maybe one or two genes in the other mice was different than the rest of the five mice, so that mice was not able to efficiently take up the drug. Some things like this is just an example. So things like this are more common in case of non-syngenic mice, where the genetic makeup also plays a very big role. But in case of your syngenic mice, if you do it in six mice, maybe the level of shrinkage of your tumor might vary, but the drug has to affect all your six mice tumors. So in case of your syngenic mice, all the mice will show the effect. The variability might be in the level of shrinkage of the tumor depending on the size of tumor and all this, but even there you should get your significance. If the shrinkage is 50% in one mice, in the rest of the mice also it should be plus or minus 2, 3 or 4%. That's it. It cannot be like 80% in one mice, 10% in one mice. Then you won't get statistical significance. This is what is reproducibility and statistical significance in case of mice. So syngenic mice model are very reproducible. That is, most of them will give a very similar response. If all the rest of your steps and protocols and your drug delivery, drug injection has been done similar way. If the handling, the delivery of the drug was not done in a similar way, then again several other factors will uh, play role. That's why whenever you're giving one treatment, you should always see that when you're doing replicates or when you're doing n is equal to 6, all of them should be given at the same time to all the mice. Everything should be exact concentration. Everything should be given in the same way, either intraperitoneal or direct injection or oral, whatever way you're giving the drug. So for all the six mice in a group, you have to follow the same protocol. Then you will get statistically significant results and with reproducibility. And reproducibility also, if one lab has done some experiment, if someone wants to replicate the same experiment and check for something else, so if they have also used the same drug, if your drug in your mice model, if it has shrunk the tumors, if some other lab is doing with similar with the same strain of mice with the same drug, in their case also the drug has to shrink the tumor. So that is also reproducibility. So whatever you have done, if some other lab or some other scientist is trying to replicate that, it has to replicate, otherwise there is some issue in your experimental setup. Okay. So then next is controlled tumor studies. In syngeneic tumor models, tumor cells derived from the same genetic background can be implanted into the mice without rejection, enabling the study of immune response to cancer and immunotherapy effectiveness. Similar, then standardized disease modeling. Inbred syngenic mice ensure that all mice respond similarly to diseases, just what you were discussing, even in case of diseases, not only in case of your immune system response, etc. In case of diseases, in case of infection, treatment, in whatever experiment you're doing, they all will respond similarly because of their genetic makeup, making it easier to attribute the effects to the intervention rather than the genetic differences. So, predictable pharmacokinetics and drug responses. Because genetic variation is minimized, drug metabolism toxicity profiles are more uniform, improving the reliability of preclinical testing. So, once your drug efficiency is checked, we should also do your pharmacokinetic studies, and you should do your toxicology experiments, and your how your drug is being metabolized in the system. So all these are called as admit properties of the drugs. So all these things have also should also be checked after going into the clinical trials, before going into the clinical trials. So there also, even how after the function is done, or how after delivery of the drug, how your body reacts to the drug directly before the drug can affect the target cells or target, how your body recognizes the drug, how it digests it, or how it handles your drug also should be similar in all the mice. and how your drug is later metabolized and released from the host system. After the function is done, the drug has to be eliminated, right? Whatever medicines you take also, once its function is done in your body, it has to be eliminated from your body. So even this method of elimination, the metabolic pathways that are involved in this, all this should be similar in all the mice that you're using. The uniformity should be there. And toxicity profiles, the drugs which you use is against some disease or against some tumors, so this drug at the same time should not be toxic to your own cells, to your own healthy cells. So in case if your drug is affecting some tumor, but at the same time it's also having some side effects, all these things are also studied for your preclinical studies and your clinical trials. So for all these kind of side effect studies and all also, the response that the host system gives should be uniform, and this should not be intervened by genetic variation. So that is the reason the syngenic mice model are very useful. So this helps in improving the reliability of preclinical testing. Then syngenic mouse model are widely used in cancer, immunology, and transplantation studies. So this are examples of your syngenic mice, inbred mice. So AJ mice, high incidence of spontaneous adenomas. So these mice also are used in a lot of tumor studies in your adenomas, hepatomas, etc. Then your C3H, HEJ, your C57 black 6J mice. This is one of the very uh, widely used mice model. So all of them homozygous across the entire genome. All mice within a strain are genetically identical, reproducible models for diseases. These are the main points that we have to remember about the inbred mice and syngenic mice. Next, single knockout and double knockout. So knockout is basically inactivating a gene. So in your single knockout, a single gene is inactivated in the entire genome. And in the double knockout, two different genes are inactivated in the organism. So in the same organism. So why do we do single knockout and double knockout? Is single knockout we use to study the effects of the lack of the gene, losing that gene in the biological process and diseases. So if you want to study what is the importance, what is the role of that gene, either in general biological physiological condition or in some disease condition, or in a particular pathway if it is involved, so you can knock out that gene and then see how your pathway is different or how the response is different, what is the effect. And in case of double knockout, it is generally done to study the interaction between two genes and how their combined loss affects the organism. So uh, if you want to check uh, two interacting partners in a signaling pathway, if you want to check whether uh, the genes are giving a cumulative effect, or the genes are showing uh, counter effects to each other, or if they are, whether they are actually interacting or whether they are just complementing each other's role, all these things can be done with knocking out those two genes. Then example for signal knockout is a mouse lacking P53, which is important in tumor suppression, helps researcher understand its role in cancer development. So when you knock out one specific gene, that is your P53, you will know the role of that particular gene only. But in case of your double knockout, you will have a knockout of P53 and RB. RB is downstream in P53 cycle pathway. So your P53 and RB double knockout mice, both tumor suppressor genes are knocked out. So this will provide insights into more aggressive cancer research when compared to single knockout mice, because you have several tumor suppressor genes, and P53 and RB come under in the same pathway. So your P53, P21, your RB. So you can study whether a single gene is affecting or not, but that will not give you more details into the entire pathway. But in case of your double knockout, you can get more details into more specifically how that entire pathway is affecting in case of cancer, because both of them are tumor suppressor and both are in the same pathway. Then why use double knockout instead of single knockout? So redundancy. Some genes compensate the effect of each other. Knocking out both can reveal hidden effects. So like I said, for interaction studies, so we can see whether two are interacting, whether two are doing the same function, two are doing a function that is opposite to each other. So in case of some genes, they will compensate each other's function. So this kind of effects can also be studied in case of double knockout, because when you do single knockout, maybe gene A you have knocked out, but there is some other gene X that is doing the same function. Maybe you want to check absorption of glucose or absorption of any kind of metabolites. You think knocking down of gene A will stop this absorption, but there is also gene B which will help in the absorption of the same thing. So you will not see any effect because your whatever metabolites you're seeing is being absorbed. Then you can find out there are some other genes also that is playing part in this function. It's not one gene effect. Okay. Then pathway analysis helps in studying complex biological pathways where multiple genes interact. Like I said, so for a single function or a single effect, it's not always that only one gene is responsible. It can involve several genes, several pathways, several steps, and sometimes it will be a complex of proteins that are doing one single function. So the whether the lack of one single gene is important or the lack of two genes is important in that biological pathway can be known in double knockouts. So mostly all the biological pathways are a network. So there are several genes, several proteins that are involved. So you can always go for double knockouts to find out the gaps and fill in the pathways. Then disease models more accurately mimic human diseases that involve multiple genetic mutations. So, in case of many syndromes or in case of neurodegenerative diseases where mutations are the cause for the onset of the disease disorder, and in case of cancers where the oncogenes are mutated. So in these kind of models also double knockouts help, because it's not always a single gene mutation. Sometimes more than one gene mutations will result in this kind of disorders and cancer. So you can mimic that effect in your disease models where you can do double knockouts. If two were involved there, same two genes you can inactivate here and then try to see how to compensate this effect, etc. The primary advantage of creating single knockout. Now, why single knockout is advantageous before double knockout is that uh, it allows researchers to understand individual gene function. By studying the effect of knocking out of a single gene, researchers can determine its specific role before investigating interactions with a second gene. So, why single knockout is important before double knockout? Now we are discussing. So, if you want to study the interactions, you can definitely go with double knockouts. But before that, you should know what each gene role is. So first you can do single knockout, know the individual effects of that gene, whether they are same, they are different, whether they are combined. Then you can do double knockout to see whether they are interacting or not interacting, or it is some indirect pathway that is giving a combined effect. So you can always go with single knockout first. Then to avoid redundancy issues, some genes have compensatory mechanisms. If a double knockout is made first, it may be unclear which gene contributed to the observed phenotype. So same in case of the previous one also, this redundancy issue was there. So in this also, so in some genes the compensatory mechanism, you will not know which gene is affecting the phenotype. sometimes the compensatory effect might not be equal. It might not be that both of them always have the same function. Sometimes it might be because the other gene is not functioning, this gene is trying to compensate it, or generally the function is something else. So first, if you do a single knockout and then do a double knockout, this kind of redundancy issues can be avoided. Next is establish a baseline phenotype. So single knockout mice provide a reference point, make it easier to interpret how additional gene deletions in the knockout mice alter the phenotype. So if it's a cumulative effect, or if it is because of several genes or two genes also, so one single knockout gene will give you a baseline phenotype. Then you can alter your second gene and see how that baseline phenotype is getting altered. So this kind of variation studies also, this is little deep into how you can use this single knockout and double knockout first and second to study different setup methods. Then, to reduce complexity and cost, generating and analyzing single knockout is more straightforward and less expensive than immediately producing double knockout mice. This is uh, obvious. And identify lethal mutations. Some gene knockouts may cause embryonic lethality. By testing single knockouts first, researchers can determine whether a gene is essential before attempting double knockout. So basically, what is suggested is before going for two genes together, if you go with a single knockout first, then you can have several reference points, several standards that can be set. You know, you can maybe avoid a double knockout if it was not necessary in the first place, or you can make the double knockout with much more information because of your single knockout first. And if your knockout is lethal, you need not, you know, spend so much time and money to go for double knockout if your single knockout itself is lethal. So these kind of things little advantageous if you go with single knockout first. Okay, so this is the basic process of knocking out. So how it is done is isolation of embryonic stem cells is done, and you have your vector, you have your gene of interest which has to be knocked down, or which has to be knocked out, silenced. So your gene X with targeted injection. So homologous recombination will occur from your vector, and now your gene is disrupted. So now this gene has become non-functional. So the inactivation of gene X, that is gene of interest, and incorporation into the embryonic stem cells by electroporation. So this gene will now be incorporated into the cells. Then isolation of the knocked out embryonic cells. So when the incorporation is done by electrophoresis, maybe not all of the cells will take up the gene. So we have to have a selection media in which the cells will be selected only when the gene of interest has been taken up, so that you will avoid heterogeneity here. So only the cells that have taken up your knockout, that is your disrupted gene, so those will be selected, and then insertion of your knocked out embryonic cells into a blastocyst will be done. Then implantation of this blastocyst into a surrogate mother, and then you will get a chimeric mice. So because this implantation is done in the surrogate mother, you will get a chimeric mice where uh, in which both expression will be there, knockout and normal. uh, and then chimeric mice crossbred with wild type mice. So now when your chimeric mice, that is your heterozygous, will now be crossbred with your wild type mice, again you will get different types. So out of this, again interbreeding of heterozygous mice will be done. Now when you get two heterozygous mice, you will interbreed them together, and then here you will get a homozygous knocked out mice. So now this is your mice model of knockout, complete knockout. So this is how a knockout mice is generated. So you can see how many steps are involved. So that is the reason first doing a single knockout is better and then going for a double knockout, because if this phenotype affect whatever is altered or unaltered, or whether it is lethal, if it is known, then you can decide whether how to go ahead with your double knockout or how to alter your double knockout study also. Okay, so that is done. Now we have density gradient centrifugation. So density gradient centrifugation is a widely used method in immunological research for separating different population of cells, organelles, and also other biological particles. This is done based on their density differences. The technique involves spinning a sample in a centrifuge, creating a gradient of density within the medium, usually a solution of sucrose, percol, or any other density medium substance, and then isolating the fractions that are distributed according to their density. So, density gradient centrifuges, as the name suggests, based on the differences in the densities after centrifugation at different time points, at centrifugation at different speeds, help us isolate cells, organelles, and other particles of different densities. Because every particle will, once density gradient is set up after centrifugation, your sucrose or your percol or whatever is there, it will settle in a decreasing or increasing density gradient. So each particle or each cell, it will go and settle in the area to its similar density. So that is how different cells with different densities will be separated. So the basic principle is gradient creation. A gradient is a different uh, densities, like 5% density, 10% density, 20% density, 30% density. This is a gradient. A density gradient is created within a tube, often using a stepwise or a continuous gradient of a dense solution like sucrose or percol. So sucrose and percol are the main density gradient solutions we use, because these are very dense solutions, highly dense solutions, and a density gradient can be created in this. This allows the separation of particles based on their buoyant density. Buoyant density is the density in which it will remain in a suspension. Then the particles, cells or organelles, whatever you want to isolate, will migrate through the gradient during different centrifugation steps and will settle at a point where the density equals that of the surrounding medium. Like I said, it will go and settle at that percentage of density where its own density is similar. Then centrifugation, the sample is placed in a tube. The tube is subjected to high speed centrifugal force. So due to the centrifugal force, the particles with different densities will separate at different points, that is at different time in the gradient, with the densest particles migrating to the bottom of the tube and the lighter ones staying at the top. So if you go from the bottom of the tube, your density will be decreasing. So whatever is the highly densest will go and settle at the bottom of the tube, and slowly the density starts decreasing as you reach the top layer. Then fraction collection. After centrifugation, the gradient can be fractionated. Usually you can do it directly with a pipette. You can collect each layer. You will get whatever is suspended in that particular layer. And then the different layers, which represent different populations of particles, either cells or organelles, or even bacteria, virus, etc., can be collected separately for further analysis. So you can see here, this is a blood sample. So whatever I explained there, here they have used Ficol, PIC, and this is PBS diluted blood. So before centrifugation, this is how it looks. After centrifugation, everything separates from each other based on their densities. So first you get your plasma, that is your top layer is your plasma. Then you get your PBMCs, then you get your fecal plaque, and then you have your erythrocytes and your PMNs. So you see whatever is the highest density, Your erythrocytes or your PMNs had settled at the bottommost part of your tube, and then you have lighter density, which is lighter than this layer. You have your fecol here, then you have your PBMCs here. You can see a fudgy kind of separation layer, that is your PBMCs, and then you have your least density, that is your plasma. So this is before the centrifugation, this is after centrifugation. What are the application, how else can you use this, is isolation of specific cell populations. Density gradient centrifugation is commonly used to isolate specific populations of immune cells from whole blood or tissue samples. So previously what I showed you, this was from your whole blood, and then you can also do it in your tissue samples, because your tissues contain different types of cells. They have different your epithelial cells, your erythrocytes, your leukocytes, your lymphocytes, and different tissue itself will have many types of cells within them, and each cell will differ in its density, so you can make use of that and separate them from each other in different layers. So this is specifically useful in studying immune responses or understanding disease pathogens. For example, mononuclear cells. So your T cells, B cells, macrophages can be isolated from blood using gradients such as Fecol-Pake or Percol, which separates mononuclear cells uh, from the granulocytes and erythrocytes. Then separation of subsets of lymphocytes can also be done. The technique is particularly useful for isolating specific subsets of lymphocytes, such as T cells, B cells, or dendritic cells from the blood or the lymphoid tissues for functional and molecular studies. So based on the type of the density medium you use, based on the type of density technique centrifugation technique you use, you can separate as much as cells you uh, want from your tissues or your blood samples. But you should see that your difference in your cells density must be there in and your media, your percol, fecol, whatever you're using should be compatible to that difference. Then separation of granulocytes and monocytes. In immunological studies, the method can also be used to separate granulocytes, that is your neutrophils, eosinophils, from monocytes and other immune cells based on their different densities, allowing the studies of specific cell functions behavior. Once you isolate, you can then definitely study individual cell functions, individual cells behavioral function, etc. Purification of organelles or vesicles. So not only cells, like we said before, the organelles within the cells can also be centrifuged, because every organelle has a different density. So based on this, they can also be separated after lysing your cells. So beyond cells, density gradient centrifugation can also be used to isolate intracellular structures such as endosomes, lysosomes, exosomes, which are important in immune signaling, antigen processing, and immune cell communication. So if your studies include intracellular organellar experiments, like if you want to separate mitochondria, you want to separate ER separately, endosomes separately, you can also, separate them, isolate them, and then use them for specific studies. Exosomes are also used for delivering drugs, delivering certain thing, biomolecules into the cells. So for all this also you can make use of density gradient centrifugation to isolate the intracellular structures such as your endosomes, lysosomes, exosomes, even your mitochondria, etc. Then purification of antibodies and other proteins. In some immunological research, density gradient centrifugation can be done to isolate antibodies, antigen antibody complexes, or other proteins, which are important for studying immune responses or developing immune therapies. So it can go to more distinctive separation as distinct you can make with your sophisticated centrifugation technique. So from cells, from blood, plasma, erythrocytes, lymphocytes, you can even go up till proteins like antibodies, antigen, antibody complex. Even to that specific level you can go and separate them because of the difference in the densities. Study of pathogens. The technique can be used to isolate pathogen infected cells or virus particles. For example, virions. These are virus particles can be separated from the host cell debris using density gradient centrifugation. So now, after infecting the cells with virus, if you want to then again isolate the virus particles, that also can be done. Then or from a sample, if you want to isolate the virus particle, from a sample you want to isolate your pathogens, that can also be done. So the centrifugation, this can be done to study your structural details of your pathogens, your viral structure, infectivity, or to isolate RNA, DNA again for further analysis. Then the common types of density gradients that are used are your fecal PEG that we just saw, a commonly used solution for isolating mononuclear cells, that is lymphocytes and monocytes, from whole blood. It has a density that allows the separation of lymphocytes from red blood cells and granulocytes. Then we also have Percol. This is a silica based solution. This is used for separating cells or organelles, your mitochondria and all these organelle level. You can use Percol. So that is based on what you want to separate, you have to choose your density gradient media. So especially higher resolution is required for certain applications. You can go ahead with Percol. Then sucrose or other sugar gradient. Sucrose is a very general density gradient that is done. These are used to isolate subcellular organelles like mitochondria, endosomes in studies of cellular function and immunology. So based on the type of experiment you want, based on how you want to use your isolated organelles or cells or your virus or pathogens, you can choose your specific type of density gradient medium that is compatible to it and that is more helpful in further downstream processing. So this you can check with the protocols. Each medium will have a slightly different protocol. So based on how you want it, what do you want to use your isolated cells and particles, you can go ahead with any of these methods. So you can see here, this is from your cells. Uh, this is your 300G, this is your mixture first, then you have your 300G centrifugation for 10 minutes, then you get your cells at the bottom, then whatever is lesser density still remains at the top. Then further, if you then you can collect this top part and you can use this bottom part if you need the cells, or if you want further isolation you can collect the top part and go ahead for further centrifugation. Then further for 2000 G for 10 minutes you will get the dead cells settled at the bottom, then you have the remaining here. Again you can collect the top part and centrifuge for higher speed, that is 10 G. You see at every step to isolate a lighter density we have to go with a higher centrifugation speed. So here again we get the cell debris, then you have something remaining here. Then again when you increase the speed, now we increased it to 100,000. So now we get rough vesicles that were within the cells. Then again if you centrifuge it for 100,000 G for 90 minutes in the previous suspension, then again you get some rough exosomes, etc. Then you can see this one again you can go for further isolation. So you can see how each further and further smaller particles you want to isolate, your complexity and your centrifugation speed, your time increases. This can again be subjected for suspension, and then further increased centrifugation can be done with density gradient. So here you can see how your density gradient helps in smaller substances. So now once your density gradient is created, you have your bottommost highest density 90%, then you have your 45% layer. This will depend, this gradient will depend on your media that we use, which will separate at what gradient. Then you have your 35% gradient layer, then you have your 21%, then you have your 5, then the least dense will remain still on the top. So here whatever mixture is given here after centrifugation based on its density, whatever proteins were there in this particular mixture, proteins were of highest density, so they have settled in their equivalent density, that is 45%, and the remaining whatever smaller exosomes or MVs etc were there, they settled at a lesser dense layer, that is 21%. So here nothing more is very lighter. So whatever particles we need, you can collect each layer separately, and you can then further process or further isolate these exosomes and MVs also. Okay, so next is natural killer cells. Natural killer cells play a crucial role in the innate immune system by rapidly identifying and destroying virus infected cells and early stage cancer cells. So, your natural killer cells are basically for your virus infected cells and your early stage cancer cells, acting as the first line of defense against pathogens and tumors due to their ability to quickly recognize and eliminate the cells lacking self markers. So, self markers here is important. So, in the previous session, we discussed how cell identification of self and non self is very important when your T cell or your B cell or your immune cell selection occurs, so that you can also avoid your autoimmune diseases, and you can uh, effectively attack pathogens and non-self cells or altered cells such as your tumor or cancer cells. So they have the ability to recognize and eliminate the cells lacking your self markers or your self uh, proteins on your cell surface, essentially providing immune surveillance against potential threats without the need for specific antigen recognition like other immune cells. So, in case of other immune cells, we have this antigen presenting cells that that are involved, which will help present the antigens to the immune cells, the processed antigens. Then these immune cells will get activated accordingly, and the immune cell response will get activated. But in case of your NK cells, they do not need this antigen recognition. They will go ahead with the self markers. So they will directly eliminate the cells which do not have self markers. So this will help in removal of all the unnecessary cells from your body. Then key functions of NK cells is killing infected cells. So NK cells are primarily killers of virus infected cells, recognizing them through the down regulation of MHC class 1 molecules on the infected cell surface, allowing NK cells to target and destroy them. So how your pathogen infected cells get recognized by your NK cells is they are your own cells. They will also have your self markers, your MHC class molecules, etc. But due to the invasion or due to the uh, infection of your virus, these MHC molecules will be down regulated, which will help your NK cells to identify these cells that they are infected, and now your NK cells will target these cells and destroy them. Then in case of tumor surveillance, NK cells can detect and eliminate early stage cancer cells that exhibit reduced MHC class 1 expression, contributing to tumor immune surveillance. So in case of your tumor cells also, because of modifications, because of mostly all the cells are differentiated cells in your body, but if the differentiated cell then transforms, EMT occurs, or it becomes some other cell, other type of cell, it gets modified, then your self markers, your MHC markers on it will get reduced, and then this will help your NK cells detect them as non-self or altered cells, and it will contribute to tumor cell immune surveillance. Then comes cytokine production. Besides direct cell killing, NK cells can secrete cytokines like interferon gamma, that is your IFN gamma, to further activate your immune cells and enhance the immune response against pathogens. Like uh, like many other cells in your body, your NK cells also secrete cytokines at the site of infection or injury, and the particular cytokine they secrete are IFN gamma, that is interferon gamma, which will now again activate your other cells, other immune cells, to amplify your immune response or specifically activate particular immune cells against particular pathogens. So this is about your NK cells. They are crucial for destroying virus infected cells and cancer cells. They kill the cells by identifying the cells that lack your non-cell cells that lack your self markers on them, and your virus infected cell or your tumor cells, they have down regulated MHC class expression on their cell surface. So this is identified by your tumor surveillance system, that is your NK cells, and these cells are destroyed and eliminated. Other than this, they also have uh, cytokine secretion or cytokine production, where IFN gamma is released to further activate other cells to act upon or amplify the immune response. Then missing self recognition. So NK cells have receptors that recognize the absence of self markers, your MHC1 molecules, triggers their activation and cytotoxic activity against abnormal cells. That is what how it recognizes the self markers is through its uh, self marker recognizing receptors. Then placental function. Specialized NK cells are present in the placenta and play a role in the implantation during pregnancy. Specialized NK cells during the implantation process also play a role in the placenta. Then key points about your NK cells are part of the immune system. Unlike adaptive immune cells like T cells, NK cells do not require prior exposure to specific antigen or antibody. So like your innate immunity in your T cells or even your antibody production in your B cells, there is this first exposure and then your second exposure. So in the first exposure, memory is created, T cell memory, B cell memory is created uh, in your adaptive immune system. So in case of your innate immune system, this is not required. They immediately identify and destroy the cell, because their major destruction recognition marker is your self cell markers. So if self cell marker is not there, it will destroy it. But in case of your adaptive immune system like your T and B cells, they require your antigens, which will help uh, recognize and generate antibodies or generate cytotoxic functions against that particular antigen, which require a memory and which require a prior exposure. Then this response is rapid because it's innate immunity, it is rapid, it doesn't need the prior exposure, it doesn't need memory, so it immediately responds and it's very quick. So NK cells can quickly respond to the threats and initiate the immune responses without any delay. Then regulation by inhibitory receptors. So NK cells also express inhibitory receptors that interact with MHC class 1 molecules on healthy cells, preventing them from attacking the normal tissues. Like in the previous slide, we read about the regulation of this neutrophils. So their macrophages uh, help regulating them. They stop the neutrophils from acting, and then they remove the neutrophils from that area once the damage control is done. So in the same way, NK cells, so that is done so that the neutrophils do not attack your healthy cells. So in the same way, NK cells also have regulatory mechanism, that is the same your self markers, that is your MHC. So then after killing all the cells which do not have MHC markers, it will still detect the cells. Then when it comes in contact with the cells which have your self markers, then it will stop its killing function. It will stop producing the hyphen gamma. It will stop the inflammatory response to the cells, because if it continues, it will also damage the normal tissues. So this is your NK cell. These are the several ways how it acts. Then cytotoxicity, it can also activate your macrophage. Then it releases hyphen gamma TNF to activate the macrophage. Then it can also activate your T cells through hyphen gamma. Then hyphen gamma and TNF also activate your dendritic cells. Then in turn, the dendritic cells and hyphen gamma also again interact with the interleukins with your NK cells. So all these signals, so all your immune cells, all the immune system, they interact, communicate with each other, and conduct the immune response against any type of damage or any type of pathogenic invasion or infection or in disease conditions. So this communication method of activating several immune responses or amplifying the responses are also done by specific cells by release of several cytokines, chemokines, etc. So this is how your NK cells attack your cells and regulate the immune response. Now coming to the dendritic cells. So the dendritic cells are derived from bone marrow hematopoietic stem cells. They are a part of a unique lineage, a unique hematopoietic lineage distinct from other leukocytes. So how do dendritic cells develop? Basically, they develop from common dendritic cell progenitors. Then they also develop from multipotent lymphoid early progenitors, MLPs. Then they also develop from DC precursor downstream of the common myeloid progenitors, CMPs. They develop from lymphoid primed multipotent progenitors. So these are all the lineages from which dendritic cells can be derived. Then what are the factors that influence the DC development are cytokines like GM-CSF and tumor necrosis factor alpha. All these enhance the differentiation of the DC cells from bone marrow precursors. Then IRF8 transcription factors regulates chromatin at the lymphoid primed multipotent progenitor stage to induce early commitment towards DC cells. So these are particular things in the developmental stages. So this is just a basic. If you go deep at every step, every progenitor lineage, what factors induce? So all these transcription factors, GM, CSF, at every step there has to be a stimulus which will help it differentiate into something. So that is how the cells get primed, the cells get differentiated into one specific type of cell. So your FLT3L strongly expands your progenitor DCs to your DCs in vivo conditions. So we can also make use of this system in your in vivo and in vitro studies if you want to, check the activation of some cells, or in developmental stages you want to check which lineage is giving rise to what cells. If you alter something, if one cell can develop into a different type of cell, if a progenitor cell can give rise to two different types of cells based on different stimulus it receives. So these kind of studies can also be done using this information. What is the role of dendritic cells? Dendritic cells are vital for immune response, specialized to take up, process, and present antigen. So basically, they are antigen presenting cells, and they have the capacity to stimulate the resting T cells in the primary immune response. So they activate your T cells. So this is your lineage from which your dendritic cells are derived. Basically, your multipotent hematopoietic stem cells. So you from there you get your myeloid precursors, you get your lymphoid precursors. So these are all markers to specify the type of cells, like you have your T cells, your CD4, CD8 markers, etc. In the same way, you have your FLT3 negative cells in your myeloid and positive cells in your lymphoid and myeloid precursors. Then again, from there, with specific stimulus, you get your erythrocytes, megakaryocytes, granulocytes, macrophages from your myeloid, and from your lymphoid, you get your T cells, NK cells, and B cells, and from both myeloid and lymphoid you get your conventional dendritic cells and also plasmacytoid dendritic cells. So this is from Nature Reviews. If you want to know more details into lineages, you can also look into several reviews which explain this very clearly. Okay, then next is macrophages. Macrophages are immune cells that play a crucial role in the body by acting as first responders to infection and disease and tissue damage, primarily through phagocytosis, that is, engulfing and destroying of the foreign particles, bacteria, dead cells, and cellular debris, while also initiating immune responses by presenting antigens to other immune cells and contributing to the tissue repair and homeostasis. So your macrophages are basically your phagocytic cells. They are present in the monocyte first, then upon stimulation, they are present as monocytes in your circulation. Upon stimulation, they get activated and turn into macrophages. So once they turn into macrophages, they will go and engulf your pathogen, or they'll go and engulf your any kind of cellular debris, dead cell, anything that has to be cleared from that area, foreign particles, and it will phagocytose it. It is also a antigen presenting cell, APC, that it is both phagocytic cell and a APC cell, that is your antigen presenting cells. So it can present after digestion of the foreign particles, bacteria, pathogens, etc. It will digest the cell, the proteins, and then the antigenic parts of the pathogen will be then presented to other cells so that they can recognize it. It can again give rise to your adaptive immunity. It can your body will now be ready for next invasion. So it also helps in tissue repair. So you have your M1, M2 type of macrophages, then you have, that is your pro and anti-inflammatory. So once your cellular debris, your damage, your pathogen, everything is cleared, now whatever damage has happened in that area, that has to be repaired. So your macrophages also help in repairing your tissues and maintaining your normal body homeostasis, cellular homeostasis. They are considered key players in innate immunity and can adapt their functions based on the local environment they reside in. So, phagocytosis, antigen presenting, inflammation regulation. So, phagocytosis primary function is to engulf and digest pathogens, damaged cells, and debris through phagocytosis, effectively cleaning up the body tissue. Whatever dead, damaged, decaying things are there, everything will be cleared. Then antigen presentation, after engulfing foreign particles, macrophages process present the antigen on their surface, which allows them to activate other immune cells like your T cells to mount a targeted immune response. That is what we discussed. Then inflammation regulation, macrophages release various cytokines and chemokines that can either promote or dampen inflammation depending on the situation, contributing to the recruitment of other immune cells to the site of infection of injury. So that's what your pro and your anti-inflammatory. So it also does the clearing, it also does the phagocytosis, antigen presenting, etc. At the same time, it can also regulate it. If it has generated a signal of chemokines, generated a signal of cytokines to recruit your T cells or your other immune cells to activate the response, it will also dampen the signals later to stop the action of these cells, so that now your tissue can be repaired after the infection injury has been cleared out. That is your inflammation regulation. So then your tissue repair and remodeling. In response to tissue damage, macrophages can switch to a reparative phenotype, producing growth factors, other molecules that promote tissue regeneration and wound healing. So now they will produce molecules and growth factors and other molecules important for tissue regeneration. Now it has to regenerate and the wound has to be fixed, it has to be healed. Then the immune surveillance, they act as sentinels within tissues, constantly surveying for potential threats like pathogens or damaged cells. So, as I said, your monocytes are always in your blood circulation, and then you also have your tissue resident macrophages. So, these are all always under survey. These are all always patrolling. So, sentinels is like guards. So, they always guard your cells, your tissues. They keep, looking out for anything that is foreign, anything that is pathogenic, and immediately attack if something is a potential threat. Then major points about macrophages are heterogeneity. Macrophages are not a single cell type, but rather diverse population with different phenotypes depending on the tissue they reside in and the stimuli they encounter. So like I said, there are also tissue resident macrophages. So depending on the tissues they are present in, whether your brain or your liver, your kidneys or your lungs, every macrophage is a diverse set of population. Then you have your M1 M2 polarization, that is your M1 M2 macrophages. So macrophages can be categorized as M1 that are pro-inflammatory and M2 that are anti-inflammatory depending on their functional state. If the function there required is to clear your pathogens, clear the foreign materials, injury, clear the injured cell, dead cell debris, your pro-inflammatory will be activated, it will clear it up, and after it is done, your pro-inflammatory will be suppressed, then your anti-inflammatory will start and it will start your wound healing, tissue repair, etc. Then tissue resident macrophages. Many tissues have specialized populations of macrophages that are permanently located there and play specific roles in maintaining tissue homeostasis. Examples are your microglia, Kupffer cells, alveolar macrophages, etc. So these are named based on the area they are present. So microglia is present in your brain. These are your macrophage cells in brain. Your Kupffer cells are macrophages in liver, and alveolar macrophages are your macrophages in the lungs. So this is your basic difference between your macrophage pro-inflammatory function and anti-inflammatory or pro-resolution function. So your pro-inflammatory is involved in leukocyte recruitment by your CXCL8-1 release, and then activation of your vascular endothelium. TNF alpha is involved in this, and along with TNF alpha, your interleukins 6, 12, IL-1 beta are involved in leukocyte activation. Then again, activation and modulation of your adaptive immune system. There is several mechanisms involved in this. Then you have your pro-resolution function, uh, where you have your production of anti-inflammatory factors, which will now inhibit the activation of your other immune cells. Then clearance of dead cells, inhibition of leukocyte recruitment, MMPs are there, and then tissue repair is activated by your growth factors, TGF beta, IGF, PDG, MMPs, collagen, etc. So here now it will produce things, extracellular materials required for repair, scar tissue formation, tissue remodeling, collagen, etc. So that is how your pro-inflammatory macrophages and anti-inflammatory, which is also called as pro-resolution macrophages, function. Okay. Then this is second. I think one slide is missing. Okay. Okay, I think the slide is missing, but basically the slide is for, yeah. So this slide is for your CD4, CD plus T cells. So your T cells have several other types involved in it based on their differentiation and activation. So you have your naive T cells, then you have your stem cell memory T cells, then you have your central memory T cells, your resident memory T cells, then effector T cells and effector memory T cells. Then you have your uh, gamma delta T cells and your natural killer T cells, that is your NK T cells. Then you have your two types of other main T cells, that are your helper T cells and your cytotoxic T cells. So the major differentiating factor in this is your CD4 plus cells or your helper T cells, and CD8 plus cells are your cytotoxic T cells. Then again under your helper T cells also you have again TH1, TH2, TH9, 17, 22, depending on different markers and different types. And then you also have your Tregs, which are your T regulatory cells. Similarly, in your cytotoxic T cells also, which are your CD8 positive cells. So here again with further differentiation, you have your TC1, TC2, TC9, and your TH17 also uh, functions both as helper T cells and cytotoxic T cells, and also your Tregs, which are involved in both. Then next is the receptors that are involved in your allergic reactions for activation of your mast cells and basophils. The receptors that are primarily involved in activation of mast cells and basophils during allergic reactions are the high affinity IgE receptors, that is Fc epsilon R1 receptors. When an allergen binds to IgE antibodies that are already attached to a Fc epsilon R1 receptor on the surface of a mast cell or a basophil, it then triggers a cascade of events leading to the release of inflammatory mediators like histamine, causing allergic reactions. So this is how your mast cells and basophils react in your allergic conditions, because they have Fc epsilon R1 receptors which are then attached to the IgE antibody when the allergen binds to it. Then they, once it is activated, now they trigger a event, cascade of events where they now release your histamines, which are your inflammatory mediators, which will then further generate downstream allergic responses. Then your major things about this is it is specific to IgE antibody. Then IgE receptor, this receptor specifically binds to immunoglobulin E. Cross-linking activation. When multiple IgE molecules are bound to Fc epsilon receptor, then are cross-linked by an allergen. Then it triggers the activation of the mast cell or basophil. That is, multiple IgE molecules that are bound to the receptors. They cross-link by an allergen. So then the response is triggered. Then role in allergic reactions. The activation of Fc epsilon R1 is central to the development of allergic reactions like hay fever, asthma, and also in anaphylactic reactions or anaphylactic shocks. Then this is how the activation occurs. So this is type 1 hypersensitivity example. So this is your allergen, this is your antigen presenting cell, then your T helper cell, your B cell, they will present the allergen. Then your B cell will then produce antibodies. These antibodies will be bound to the Fc epsilon R1 receptor of your mast cells and your basophil cells. So now to this antibody receptor that is already bound to the cell, Now, when this allergen comes and interacts, the cells will get activated and immediately degranulation process occurs. So, these granules, they contain your inflammatory mediators like histamine. This will now trigger your allergic symptoms. So this is your allergen Ig complex, or your engagement of your inhibitory receptor Fc gamma R1. So these two will inhibit this degranulation process. If only your allergen Ig complex is formed without your receptor, or your receptor is being inhibited by your inhibitory receptor, this will negatively regulate this process. So a little information about mast and basophilus on the granular content. Both are granular. So both of them have histamine, and mast cell also have heparin and serine proteases, whereas basophil has chondroitin sulfate and tryptase, and your mast cell has carboxypeptidase A, and your basophil also has carboxypeptidase A, but at a lower level. So this is a basic structure of your receptor Fc epsilon R1, and this is your antigen binding site. This is your IgE antibody. This is your receptor, and downstream you have your ITAMs, etc., etc. So this is your receptor, your IgE antibody bound to it. When antigen comes and binds at the antigen binding site, the response will be triggered. So this is how your mast cells and basophils are activated by your IgE antibody receptor Fc epsilon R1. Okay, and next is the NADPH oxidase enzyme. So the enzyme primarily responsible for the production of reactive oxygen species in neutrophils is your NADPH oxidase. So in the beginning we read in your neutrophils, they also generate ROS to eliminate your pathogens. So how that ROS is generated is because of your NADPH enzyme complex, NADPH oxidase complex. So the NADPH oxidase is a multi-subunit enzyme complex that is activated when the neutrophils encounter pathogens or any foreign or obnoxious stimuli. So it translocates to the plasma membrane and uses NADPH, that is your nicotinamide adenine dinucleotide phosphate, NADPH, to reduce oxygen. So in this reaction of reduction of oxygen, they generate super anions, that is O2 negative, a type O2 anion that is a type of ROS. So this super oxide anion can then be converted into other types of ROS, that is hydrogen peroxide, hypochlorous acid, etc. So which are toxic to invading pathogens. Then what is the function of this ROS? Is ROS are a part of respiratory burst that neutrophils undergo when responding to infections. So this is a respiratory burst which generates immediate ROS levels uh, to attack the pathogen. So they are used to kill and digest the bacteria, fungi, and other pathogens that are engulfed by the neutrophils during phagocytosis. So once the bacteria or any pathogen that is phagocytosed, immediately ROS is generated by NADPH oxidase, and this ROS will now help in digestion of this pathogen in the phagolysosome. Then overall process is essential for neutrophil antimicrobial function, though excessive or dysregulated ROS production can contribute to tissue damage or chronic inflammatory condition. So like in case of any immune response, even this has to be regulated, because excessive ROS is also dangerous to the own cell and other cells surrounding it as well. So this ROS has to be cleared from the cell after it has been utilized in its antimicrobial functions or to kill the pathogens. So now this ROS will be acted upon by your dismutase enzyme or other enzymes to clear it. So this is your neutrophil So it has a phagolysosome in which it has a bacteria that it has engulfed. So this is your phagocytosis process. So here you can see generation of ROS. So you have your NADPH here, then it is reducing your oxygen into superoxides anions, and now your superoxide anions are then being converted to your hydrogen peroxide OH and your hypochlorous acid, etc. So now you also have several genes and other pathways that are involved in it, like your NOx2, INOS, your SORD, NPO, that is your dismutase enzyme, superoxide dismutase, myeloperoxidase. So all these are involved, and it's a pathway signal that occurs. Then once the damage to the bacteria or pathogen is done, this ROS will then be cleared from the cell so that it doesn't then generate inflammation and tissue damage and cause chronic inflammatory conditions. So this is how ROS helps in pathogen clearance or antimicrobial functions of neutrophils. So that is it for this session. Uh, if you have any doubts, or if you want me to repeat any portion of it, you can let me know. Anybody, any doubts?
No, ma'am.
Uh, Ma'am, I have a doubt. Do we have to really completely study the whole flow chart of the you said NADPH oxidase for the exam?
For the exam, whatever was the syllabus is given, that is enough. You don't have to study more in detail. So this was just to, make you understand the complete thing.
Sure, sure, sure.
Okay, how much is required? Like if
Yeah,
I
that's what I'm.
yeah, whatever is in the
I'm
syllabus,
asking
that
whether
is
I have to
enough.
study the whole biochemistry of this thing.
No, entire biochemistry is not required. Basically, how it is involved in clearance of your pathogens and how it helps in your antimicrobial
Mhm.
activity, that much is enough.
Yeah, okay, sure, sure, sure. Thank you.
Okay, thank you. Anyone else?
Ma'am, just a small query. I missed the first class, I mean, first live session, and there isn't YouTube video that I can access, YouTube link that I can access. Do you have that link, ma'am,
Yeah,
for the first live session?
actually, yeah. So that session, actually, there was a error in the recording. So the audio
Oh,
was not recorded. So that's why I hadn't uploaded. But I will re-record it and I
sure,
will
ma'am.
upload it in the YouTube.
Okay, thank you.
Okay. Yeah. Okay. Then I think if anybody is not having any other question, we can close the session. Okay. If you have further any queries, you can post on the group for the course. Okay. Thank you everyone for attending.
Ma'am,
Yeah.
how to download this um, slide share, ma'am?
Uh, there is a download option, right? It is uploaded in the drive.
Yeah, yes, ma'am.
So there, if you go open it, you will get a download option. You can download it. Are you facing any trouble in that? Do you have any issues while downloading? Are you getting any errors, or what is the problem? I don't get it.